Hello everyone, I am Moksha. Welcome to Catchy Med Concepts. This is the outline of the GS signaling pathway which we'll be using to understand this topic. First, we will begin with taking a look at this receptor. Now, this is a G protein coupled receptor. It is a snake-like receptor which will cross the membrane 7 times. It is represented by the green dots. Now, as the name suggests, G protein coupled receptor. It means that it is already associated with 3 G proteins right here. They are called as the alpha, beta, and gamma. What happens is that alpha proteins in the resting phase, where it is inactive, are bound to GDP, as we can see right here. But in the active phase, that is when any signaling molecule comes, they start to lose their affinity for GDP and start to gain affinity for GTP and attach to GTP right here. Now, this whole complex of alpha and GTP goes and activates another interesting enzyme in the cell, which is called as adenyl cyclase. Once this adenyl cyclase is activated, it converts the already present ATP inside of the cell into CAMP, cyclic AMP. This cyclic AMP is basically a secondary messenger in the cell which plays a very important role. But before diving into the roles of CAMP, we will first look at the fate of this complex of alpha and GTP. What happens to this alpha and GTP is that adenyl cyclase only, once activated, converts this GTP back into GDP right here, as we can see. Okay, we have seen this arrow going into opposite direction because the action has been performed by adenyl cyclase only. Now this whole complex of alpha and GDP will go back to its original position right here, as we can see. Now talking about CAMP. What happens is that CAMP activates PK. How it does it has 4 molecules of its own which will attach to 2 molecules of pK, and each molecule of pK will have 2 units. One is the regulatory unit, one is the catalytic unit, and the regulatory unit will already have grooves in them to embed the molecules of CAMP, as we can see right here. Now these red dots are the molecules of CAMP. Earlier we saw over here that the bondage in between regulatory and catalytic unit is very high and a very tight kind of bonding. But once this molecules of CAMP is attached to the regulatory unit, the bonding will be loosened up a bit and this catalytic unit will be set free. Now after all of this, we will have an activated pK. Having an activated pK was our whole point of understanding this concept, is because this will do all the magic. pK is involved in the expression of the message that these signaling molecules were carrying by adding or removing phosphate groups, which is phosphorylation or dephosphorylation. One of the major functions of this protein kinase is also the activation of various transcription factors such as CREB, CREM, and ETF. Why this is so important is that because these transcription factors are involved in the cell proliferation, cell differentiation, cell survival, cell growth, cell apoptosis, etc., and all the processes revolving around cell. Hope this helps you understand the concept. Thank you so much for watching.